So we'll also be looking at water testing and coliforms. So you already had a question like this on one of your quizzes. What traits differentiate coliforms from other organisms? There were a list of possible traits that tell you something is a coliform, so you want to know those. And what media tests are used to look for coliforms. So for this, you want to know the full names, as well as the short names. So you know it was presumptive, confirmed, and complete. But what are the actual media that are used for those tests? And what's the purpose of each media? So they each tell you something different. And what constitutes positive results? So I can ask that question right now. It's fairly easy. When you're doing the water MPN test, what's positive?
Gas production. Right.
Okay, so one thing is gas production and growth. So it has to grow and produce gas. If you just have growth, that's a negative result. And if somehow you just have gas, that's also a negative result. And so the final thing to know for this is calculating MPN. So in class, you just used a table that you had 5 positive tubes of one type, 2 of the next, and 1 of the other at each dilution. So you calculated MPN based on that. You could just look at the table and find what your most probable number was. But you need to be able to actually use the formula. And I have that listed here, but it's also in the handout if you need to find it.
So we need to memorize that formula?
Yes.
Okay.
As far as I know, you're not getting a table. So be good to know. So for the MPN procedure, you use Laurel tryptose broth as your first test. That's the presumptive. And I'll leave you to look at what the actual media means. And then your confirmed and complete test are brilliant green and E. coli. And so you can also look up and see which one of those is total coliforms versus fecal coliforms, things like that. So we're also looking at blood agar plates, and this relates to the human flora lab. So there were 3 different results you can see on blood agar: alpha, beta, and gamma hemolysis. Alpha hemolysis is the brown or greenish color that you get. There's no clearing of the media, it just changes color, and this is partial degradation of the hemoglobin. Beta hemolysis is complete degradation, so you see this clearing on the plate. And gamma means that there was no degradation of the hemoglobin at all, and so the plate stays as is. There's no clearing or color change. So you should be able to look at those results, and based on this test and a couple others, determine whether or not you think an organism is pathogenic or non-pathogenic. So these aren't completely finished tests. So if you just do blood agar and you get beta hemolysis, for example, that doesn't necessarily mean that you have a pathogen, but that sort of puts it into that bin that it could be a pathogen. So they're not conclusive, but they help you narrow down the field. So that's what we're looking for here. Be sure to know the observations and interpretations for the various types of media that you can use to determine whether something is pathogenic or non-pathogenic, and why you're using these tests in the first place. So some of the media that we'll look at is blood agar, MSA, Staph 110, coagulase, and Baird-Parker, and I'll leave you guys to figure out what observations and interpretations relate to the pathogenic versus the non-pathogenic, and why we can use each of these tests. So for these, you only need to know their relation to human flora. So for example, blood agar is fairly straightforward, right? It's on blood, so if it's lysing or degrading hemoglobin, then that means it could be pathogenic because it can affect your own hemoglobin. If it's gamma, then that means it probably isn't pathogenic because it's not doing anything to your blood cells. So that's a fairly easy one, but the rest you guys can look at on your own. We also looked at antibiotic and antiseptic susceptibility. So for this, you just want to be able to measure zones of inhibition. So remember that those are, you're measuring the diameter across the center, and look at those on an antibiotic or antiseptic susceptibility chart and determine whether they're resistant, intermediate, or susceptible. And so just as another thought question, if you look at all these different zones of inhibition here, we can see that this is the largest zone of inhibition, but does that mean that it's the most effective antibiotic, and why or why not? So the biggest zone of inhibition does mean that there was the most bacterial killing, or if it's bacteriostatic, then it stopped the most bacterial growth for the longest distance. But what detail are we missing about the plate and the discs on it that would lead you to whether or not this was the most effective. What bacteria is on it, so that's a good point. We don't know what bacteria, so if you knew who you were looking at, say if it was gram-positive, then you would know something like penicillin that degrades the cell wall might be very effective, or if you know that it's completely lacking in the target of the antibiotic, that it wouldn't be effective at all. But what about the discs themselves? Do you need to know to determine whether the antibiotic is more or less effective than the others? So what information was on the back of the discs? The name of the antibiotic, concentration. So that's what we were looking for here. So for example, if this is like norfloxacin or something that's a DNA inhibitor, it takes a very, very small amount of that antibiotic to cause a large clearing. Whereas if you added the same amount of something like penicillin that a lot of bacteria are becoming resistant to, you would expect a much smaller zone of inhibition. So by contrast, if we said, say this is norfloxacin and it has a nanomolar concentration of the antibiotic, and this is penicillin, but it has like 1 gram per ml or something ridiculous of antibiotic, well, it's still not more effective. It did kill more bacteria, but you had to add so much that it doesn't really matter. So you want to take the concentration into account for a question like this as well. You also want to know mutations, so the calculations won't be on the practical. We don't really have the time for you guys to go through all the mutation calculations in that 4-minute time span, but they will be on the final, so this is why you want to bring a calculator. For the practical, you do want to know what data sets to use for kill rate, spontaneous mutation, and total mutation rate. So you got some good practice with this with the mutation assignment that you'll be handing in. But you just want to remember that for the kill rate, you're looking at the UV exposed versus unexposed. For the spontaneous mutants, you're looking strictly at the ones that are not UV exposed, and you're looking at your control plates against your experimental plates. And for the total mutants, you're taking into account both the UV and the experimental condition. So just be familiar with which plates you should use for which condition. Also, looking at bacterial DNA transfer, so you want to know all 3 types that can possibly be done by bacteria, and in specific, which one did we use in lab? Transduction, conjugation, or transformation? Transformation. So for transformation specifically, you want to know the difference between natural and induced transformations. So the one that we did in lab was natural. It's one that's a naturally receptive bacteria. But in a lot of labs, you have to do induced transformations. So that's things like exposure to calcium chloride, heat shock, electroporation, those types of things. So just be familiar with the methods that can be used. And also the experimental procedures that we used in our own lab transformation exercise, especially the use of controls. So I know I've been stressing this a lot, but you can expect to see it a lot, likewise on the test. So as just another thought question, you see the results shown below, so what can you conclude about your experimental procedures? And should you continue to streak this out onto another streptomycin plate, or should you start over and try again? So this is exactly what we saw in class. This is the first day of the transformation experiment, streaking just on plain NA, and you have the strep-resistant cells, strep-sensitive, the DNA from the strep-resistant, and then the strep-sensitive with the strep-resistant DNA. So which of these quadrants doesn't belong?
DNA.
The DNA, exactly. So if you had results like this, would you continue with the experiment or start over?
Start over.
Okay, good. So the other topics that you'll want to be familiar with for both the practical and the written is the plaque assays, so how to calculate the average phage per mil, microbes and their metabolic capabilities in fermented milk. So just look over the handout and the lab lecture, or the lab lecture for the yogurt class. And finally, more information on the flora of the skin, nose, and throat. So I just went over sort of the experimental side of what we did and what you'll see on the practical, but this is something that will be expanded upon a little bit more in the written. And I believe that was everything. So if you have any more questions, you can let me know. Otherwise, we can go eat yogurt.